thank you guys for coming to my thesis defense. It's such a pleasure to see you all here. The title of my defense is Engineering the Tryptophan Synthase Beta Subunit for Synthesis of Non-Canonical Amino Acids. So proteins are absolutely incredible molecular machines. They actually perform many functions that are similar to man-made feats of science and engineering, though they've existed long, long before modern technology was ever even dreamed of. However, just like machines, we found that we can harness and engineer proteins. And today I'm going to be talking about how enzymes, which are proteins that catalyze most of the chemical reactions found in the biological world, can be used to make different molecules that we as humans want to use. So more specifically, I'm going to tell you guys about this enzyme, the tryptophan synthase beta subunit, or TRPB, which naturally catalyzes the last stage of tryptophan biosynthesis using these two molecules, indole and serine. And I'm going to be talking about how it can be engineered to synthesize non-canonical amino acids. So before I can talk about non-canonical amino acids, let me first describe what amino acids are. So every protein is composed of these 20 canonical amino acids that I have listed here. And amino acids are composed of an amine group and a carboxylate group, hence the name amino acid. And each amino acid has a different functional group, which I've highlighted here in orange, that gives it different chemical properties. And amino acids can be strung together to form a chain called a polypeptide, which folds into a three-dimensional structure, which is what we call a protein. And this sequence dictates its structure and therefore its function. So different combinations of amino acids give us different structures and different functions. Now, the 20 canonical amino acids, which comprise life and all proteins, represent only a very small fraction of amino acids that could exist. In fact, there's an entire universe of amino acids beyond the canonical 20, which we call non-canonical amino acids. Now, some non-canonicals are naturally occurring, which expands nature's chemical diversity beyond the proteinogenic amino acids. For example, some organisms have incorporated a 21st amino acid into their genome, and most organisms use post-translational modifications like phosphorylation and glycosylation to affect protein function and behavior. And finally, on top of that, there's all sorts of different amino acids that are found in bioactive molecules, which are both naturally found and bioactive molecules that have been man-made, which means that there is a pharmaceutical relevance for being able to make non-canonicals. And finally, there are many non-canonical amino acids that do not occur in nature, and we call these unnatural non-canonicals. And they can possess unique functional groups that can be leveraged for basic science and also for new drug development. So I hope I've already told or demonstrated to you guys that non-canonicals can be quite useful, but how do we make them? Now, there are a few major challenges with making non-canonicals. Most notably is the challenge of chirality. So all amino acids are chiral. Well, I guess with the exception of glycine, but most amino acids are chiral. And what I mean by chirality, I'm actually, since this is such an important concept in my talk, I'm going to break it down and about what it is and why we care about it. So chirality is refers to the arrangement of atoms in space on a molecule. We also call this asymmetry. Chiral molecules are mirror images that cannot be superimposed on one another. Another important definition here of a molecule is chiral. It has a stereocenter, which we denote with a star. And molecules that are mirror images are called stereoisomers. And if there are two stereocenters, then we call them diastereomers. Now, chirality is very important because different stereoisomers can interact with the body in different ways. One stereoisomer might be an effective drug, while the other one could be toxic to your body. So it's incredibly important to be able to make selectively the particular stereoisomer that you are interested in. On top of that, all of the amino acids that exist in life that make up proteins are all one particular stereoisomer. So usually we want to be able to make that single one instead of the other one. Now, there are several ways of making non-canonicals, and it turns out it's actually fairly easy to make amino acids, but making them selectively is an entirely different story. And setting that stereocenter is usually the hardest part. In most methods for amino acid synthesis, almost the entire molecule gets synthesized beforehand, and then the last step is solely dedicated to setting that proper stereocenter. So one way we could consider making them is chemically, but this often requires protection of the reactive amine group, and oftentimes chemical syntheses are not very selective. So on top of having to protect from off-target reactions on the amine group, you may get other random off-target reactions and a mixture of products at the end of your reaction, which is not ideal. On top of that, if you were going to do this method catalytically with chemical synthesis, you would have to synthesize the catalyst potentially, which can be really labor intensive and arduous. However, one main advantage of using chemical synthesis is that most methods enjoy very broad scopes, meaning that you can actually use one chemical synthesis method to make many different non-canonical amino acids. Now, on the flip side, we could use biocatalysis. Enzymes are incredibly selective, but this can actually be kind of a double-edged sword, because what prevents off-target reactions also prevents them from having very large substrate scopes. So we often have to engineer enzymes to expand their substrate scopes. And what I mean by substrate here is it's a molecule that an enzyme performs chemistry on. And I'm going to use that term quite a bit throughout my talk, so I thought I would define it there. The other kind of downside to a lot of enzymes, not all of them, but a lot of them, is that they can perform the reactions reversibly. So what that means is that a reaction may get to a certain percentage of being done, and then the enzyme will start to break the product down into its starting materials again. So you have to be very careful with some enzymes to stop the reaction exactly when you want it, when it's completed. And finally, like I mentioned before, both methods can use expensive starting materials and may require really complex syntheses to get up to the point where you're setting the chiral center. So it sometimes makes it such that the methods are not particularly scalable or desirable. Wouldn't it be nice if there was something that maybe was the best of both worlds here? And what I'm talking about today is an enzyme that is just that. The tryptophan synthase, it is such a beautiful enzyme to work with. It performs this reaction between ethyl and serine in a single step. It's irreversible. And it is a complexity increasing reaction. So you take very simple starting materials and you make a nice complex molecule at the end. On top of that, one of the really nice benefits of working with this enzyme is that it's really highly expressing, or at least the ones that I had the chance of working with. I could grow very few cells to obtain a large quantity of enzyme to work with. And it's very thermostable, the one that I got to work with. So what do I mean by thermostable? The tryptophanes that I worked with are derived from extremophiles, meaning that they lived in temperatures around 80 to 100 degrees Celsius or 176 to 212 degrees Fahrenheit. They were called Pyrococcus furiosus and Thermatoga maritima, and they were found in hydrothermal vents in Italy. By thermostable, I mean that they have to be very stable at high temperatures, but this also confers a high degree of stability in other conditions. And it actually is exceptionally practical for engineering, because I can actually introduce a lot of mutations into TRPB before it begins to destabilize. So it's a very engineerable enzyme.
Ella, let me just interrupt for just a minute. Somebody's making background noise. Would you please mute yourselves if you're not muted already? Thanks.
Thank you. Okay, so how do we engineer new activities into an enzyme? Well, it turns out that most enzymes are what we call promiscuous. Now, what do I mean by that? So this is just kind of an abstract concept, but if we consider this is what we call a fitness landscape down here in the left-hand corner. And in orange, this first peak, we have a native reaction. And then in pink on the right side, I have a new reaction. And sometimes with enzymes, if you give them a new molecule, they have some level of promiscuity, meaning that they can accept it and perform this new reaction. So if you find this foothold, if you find this region of promiscuity, you can actually evolve on this new activity. And how do we do this evolution? Well, we use directed evolution, which Francis won the Nobel Prize for in 2018. But the way that it works is once you've found a starting activity, you will introduce mutations into that enzyme. And it's kind of a choose your own adventure process here. You can introduce mutations in a number of different ways, but you really pretty much just do what works. And if it's not working, you try a different method. But in the Arnold lab, what we do is we actually screen for new improved variants on these 96 well plates. So we array them in 96 well plates and we look for improved activity. And if you do find something with improved activity, that means you're climbing up this new fitness landscape, and you can take that variant and use it as the template for the next round of evolution. And you pretty much just rinse and repeat over and over and over again until you've reached an enzyme with the desired amount of activity that you wanted. Now, something that protein engineers often do is look at enzymes' catalytic cycles in order to imagine other substrates that it could accept or other mechanisms that it could perform. I'm going to highlight the most important parts of the TRPB catalytic cycle to help us imagine what else that it could do. So first, we have the resting state of the enzyme. It's this pyridoxal phosphate or PLP cofactor that's bound to an internal lysine. And when serine enters the active site, it actually kicks that lysine off, and we get a series of rearrangements with transamination reactions and water leaves, and we get this first and pretty much only really important intermediate that we're going to focus on. This is what we call the amino acrylate, and we have made an electrophilic species out of serine. At this point, the nucleophile in this reaction, indole, can attack the amino acrylate and make an irreversible carbon-carbon bond. And after that reaction happens, we again have another series of transamination reactions until tryptophan is released. But since the reaction is irreversible after this step here, we actually don't really care about the second half of the reaction if we want to consider anything we could change about the catalytic cycle. So I'm just going to get rid of that so we have a little bit more room to think. So another way that we could think about this reaction is, or the way that we could describe it, is that it's a bimolecular reaction, meaning that it actually has two substrates, the electrophile, serine, and the nucleophile, indole. And ideally, you could swap out the indole and serine in kind of a plug and play fashion with different electrophiles and nucleophiles in order to synthesize structurally diverse non-canonicals. And as it turns out, that's exactly what you can do with TRPB. So we've found that you can actually provide different indoles with substitutions on this different serines with substitutions on this beta position to make beta branched amino acids. And there's been even more progress with expanding TRPB's nucleophile scope. And it now accepts a relatively wide range of chemically and structurally diverse nucleophiles. So this is the current, well, when I arrived at the Arnold lab, this was kind of almost the current scope of nucleophiles that the TRPB could accept. And this is kind of an overwhelming figure, but that's kind of the point, because what I really want to show you here is that TRPB can make all sorts of things. But I'll break it down quickly. On the left side, we've got all of the different indole nucleophiles that can be accepted. So we've got different positions on the indole that can be substituted with electron donating groups, halogenation or electron withdrawing groups. We can, all the ones that are highlighted in pink can take those beta branch serines. But then here on the right, I said that TRPB could do it when I arrived, but actually, as I started my thesis work, we were all kind of wondering, we've done all of this indole engineering. What else can TrpB do? What other non-indole carbon nucleophiles could work? And my colleagues, Dr. David Romney and Nicholas Sarai, engineered TrpB to accept these nitroalkanes as nucleophiles. So they look nothing like indole. And Dr. Marcus Dick in the TrpB group also engineered it to accept these oxindoles. So as I started to think about what my thesis work would be, I was conferring with them when we were all wondering what non-indole carbon nucleophiles could TrpB react with. And we all kind of went different directions with it. So today I'm going to tell you about the direction that I went. Now, this is just the original native TrpB reaction scheme, but I wanted to have it here so that you can compare it to the different reactions that I ended up engineering the enzyme to do. So it's just, we've got a resonance structure here of indole, a nucleophilic carbon that attacks the aminoacrylate. And the first molecule that I did the engineering for was on this really cool molecule called azolene. So azolene is this 7-membered ring fused to a 5-membered ring. And because of its aromaticity, it's actually this really brilliant blue color. It's actually responsible for the bright blue that you see in these mushrooms that look like they could be straight out of a fairy tale, but I assure you they are not. They are real. It turns out if you turn azolene into the R group or the functional group of an amino acid, you actually get a tryptophan mimic that has very unique spectral properties that people really like to use for basic protein research. You can study protein dynamics and functions with this molecule if you incorporate it into a protein. Because it's so similar to tryptophan, you can actually put it in place of a tryptophan and it won't perturb the 3-dimensional structure of the protein, but you get all of these cool functional, you get all of these cool fluorescent properties that are useful. All right, so despite its utility, it actually is relatively difficult to synthesize. The best synthesis before I worked on this project was this multi-step process that had really air and water sensitive conditions, and the intermediates were unstable at room temperature. So the people who made this had to work like very quickly in order to try and get any decent yield. But because of the structural similarity to indole and the fact that we have this resonance structure that has some negative charge here on this carbon, we thought maybe we could use TrpB to make as-ALA. Now, to actually find activity, there are 2 ways that we do it in the Arnold lab. The first is that we can use just purified proteins that we have in our freezer and set up little tiny analytical scale reactions and analyze them on our analytical equipment. Or we can actually express all of the different TrpB variants that we've accrued during previous evolutions and then run the reaction in plates and then analyze it on LC-MS. So I used the first technique in this project, and I just pulled all of the ones we had out of our freezer and I tested them with azolene, and I found that most of them worked. Although I was kind of perplexed by this particular one, but I kind of wrote it off at the beginning. I was like, maybe the enzyme was dead, who knows? It doesn't really matter because I found activity with all of these other ones. It turns out that azolene actually sublimes. It just evaporates off the reaction. So I ended up choosing this TM98 star at 37 degrees Celsius as my parent of interest, because then I could prevent azolene from evaporating off as quickly as if I were to run the reaction at very high temperatures. And I realized that I could use this evaporation as kind of a feature in the screen that I developed. So what I could do is I could actually run the reactions. I put the azolene in all the wells and I put my enzymes in, and the enzymes that could actually turn azolene into as-ALA, you would have the blue color stay in the wells, and the ones that couldn't, all the azolene just evaporated off and we'd get a very clear well. So I had developed this amazing, it was just a UV screen, it was just colorimetric, but you didn't even need the plate reader because you could just look and see, oh, that was the one that was the best on this plate. So it was a fun screen. But it turns out it was such a good screen, I actually only needed one round of evolution to get a variant that had the activity that I wanted, and we call this variant TM Azul. So I did all of this work with actually my colleague and fellow Team Trip B graduate student, Patrick Almgel. And together we worked to optimize the scale-up conditions for the reaction. We optimized the purification conditions, which actually was a lot of fun because you could just track the product through all of the purification steps because it was so brilliantly blue. What I'm showing here is actually a column which is normally white, but you can actually see where the azulene is on the column coming out. And we scaled up the reaction and we synthesized almost a gram of azulene very easily in a single step. It was such a sweet scale up, and I was very satisfied with those results. Now, before we finished the paper, Patrick and I were talking about it, and we were really upset by this one random variant that just wasn't good at it, because E104 or 105, the numbering is slightly different in the two different scaffolds that I worked with. This is a really important residue. It's actually responsible, like I said, for stabilizing this positive charge that accumulates on indole. And Patrick was like, there's no way that that was a dead enzyme. It had to be something mechanistic. And so we were like, well, let's just look, let's check it out. So what we did is we looked at the E and the G mutant at this position in both the PF and the TM scaffolds. And we know that if you get rid of this for just indole activity, it's going to make the enzyme not as good because it's not going to be positioning the indole as well, and it's not going to be stabilizing that positive charge. So that is what we see. When we make that mutation, we see a decrease in activity. But when we used azulene, we actually saw that without that E104, you saw a complete abolition of activity. And we presume it's because that residue acts very similarly with azulene, but it seems to be even more important for this non-natural activity than with the native activity, which is somewhat unusual. Normally, it's kind of the opposite that you would see in proteins. Okay, so with that project done, I'm going to go into what was my personal thesis project, and that was using ketones as the nucleophiles. So again, I had finished this project and I returned back to the question of, could TrpB react with any carbon nucleophile? It's clearly we've thrown a couple of different ones at it and it seems to just take anything. And a quick Google search will make anybody realize that no, not any carbon nucleophile can be used. Some of them are way too strong and will literally explode when they come into contact with air or water. So there is some limit. It can't be too nucleophilic that it reacts with air or water. And we obviously can't have something that's so inert that it's not going to react with anything. So clearly there's some sort of spectrum that we have, and all of the molecules that we've used thus far obviously fall within kind of this sweet spot of nucleophilicity. So I just wanted to find something else that maybe fell in this sweet spot. So I looked to kind of traditional chemistry. I used a chemomimetic approach, meaning that I was inspired by what chemists can already do and asked, can enzymes do it? And there's so much literature out there about ketone-derived enolates as classic carbon nucleophiles. It's been studied forever. So the way that ketones are nucleophilic, you can have, this is base catalyzed tautomerization, but a ketone, which is just a carbon double bound to an oxygen, and then an adjacent carbon, you can get a base pulling off a proton, which leaves electron density on this carbon, which can then be kind of stabilized by an oxygen, or it's stabilized. It delocalizes that charge. And then the oxygen can pull off a proton off that base again to reach an enol. But it turns out that this enolate can also react with an electrophile. Now, chemists are very good at making enolates. They can actually make them using methods like this LDA, where they pull off that proton and then they use this positively charged lithium to just kind of stabilize that negative charge such that you have a persistent enolate that just waits around for a nucleophile to be added in to react with. But one challenge that, even though it's really easy for them to make, it's very difficult for them to control the enolate once it's been made. So it turns out, actually, despite the fact that this has been studied for 50, 60 years at least, direct asymmetric alpha CH alkylation of ketones is still an unconquered problem. There's no general method that people can use for selective alkylation. So there are 4 challenges to this. The first is that you can actually get an E or a Z enolate, meaning that this R group here that's on C alpha can either be cis or trans to that oxygen. And then the electrophile can either come from the top or the bottom of the of the ketone. And the combination of these 2 conditions can get you any of the different stereoisomers. On top of that, if you have multiple positions where the proton can be removed, you're going to have this issue of controlling which enolate gets made. And finally, once that carbon center does get made, once the carbon-carbon bond is formed, the same thing can happen. You can actually get keto-enol tautomerization again, and you get complete ablation of the carbon of the new chiral center that you just set. But I was thinking about it, and enzymes are really good at controlling these 3 things in general. So I thought maybe enzymes could be used to asymmetrically alkylate ketones. Maybe they could be a solution. So could TrpE accept them? There was one other thing that I was concerned about using enolates in water, and that's that enolates are thermodynamically unfavorable in water, meaning that they'd rather hang out as this ketone than be the enol or the enolate. And when I looked into the literature, I could not find very many examples of enolates, and those that do use them are exceptionally specialized. For example, this type 2 aldolase uses a zinc ion that's kind of similar to that lithium, where it helps stabilize that negative charge that occurs, such that this can actually perform nucleophilic attack. But TrpE doesn't have that built into its catalytic cycle, so I wasn't sure, if I didn't have the fancy machinery, if I'd be able to do enolate chemistry. But I screened for activity using this 2-fluoroacetophenone molecule. And I initially was looking for this linear amino acid, but I realized quickly that it would spontaneously cyclize to make this amine product. So I found activity. I screened the TrpE lineage plate, and I found activity for this molecule. So I performed directed evolution on it. I did 4 rounds. I actually ended up using 4 different mutagenesis methods, because those are all just what worked during that round. And I employed this method called Miser for screening it, and I reduced the screening time that it normally would take me about 5.5 hours to screen a plate, but I had gotten it down to about half an hour, and that expedited this evolution so much. I introduced 4 different mutations, and I ended up with this variant that did over 3,100 total turnovers for this reaction. And you might be thinking, Ella, why didn't you check for its selectivity if that's what you're interested in for this project? Well, these molecules cannot be synthesized chemically. So I had to evolve the enzyme to be good enough to even make this product in large enough quantities that I could purify it out and figure out if the enzyme was selective. So I did that, and it turns out that the enzyme was so exceptionally selective. And even though that I didn't do any selection for any sort of stereoselectivity, the enzyme was just really good, and it had it was selective up to about 95 to 98%. Now, I don't know exactly which one is the major diastereomer, but that's kind of beside the point. Now, the next thing that I did is I thought, okay, did I evolve generally for ketones, or was it just for this fluoroacetophenone molecule that I had used? So I tested the whole lineage against this propionophenone molecule, which has a methyl group instead of a fluoro group. And it seemed as though the evolution was not actually increasing activity for every ketone throughout evolution. And I assume this may be because I was tuning the active site to recognize the fluorine functional group, but I don't actually know. But I could hear Francis saying, hey, well, you get what you screened for, because I hadn't screened for propiophenone activity, so why should I have increased its activity? But I tested its selectivity, and here's the really cool part. It started out with about 75% selectivity for the major diastereomer, and over evolution, I had entirely flipped its selectivity. And what this is really important, or why this is important, is because we'd like to be able to use enzymes to be able to access any different stereoisomer. And so I've proven that you can use TrpB to make all of the both of the stereoisomers that we were interested in. So I visualized where all of the mutations were, and I realized that most of the mutations that I had introduced were in this active site. So this is where that PLP cofactor is, and surrounding it is where all of the mutations that I introduced were. So again, this kind of supported my hypothesis that possibly I was reshaping the active site to help guide that ketone in to be in the right position. But again, I still don't know exactly what it was doing. And I then characterized what evolution had possibly done. I looked at the kinetics of the enzymes, the starting enzyme, and then my final variant that I called TM enolate. And we see that I had this huge increase in the maximum velocity that the enzyme could reach. So this prompted me to ask, okay, what did evolution do to speed up this reaction? To answer this, we have to kind of consider, or I started to consider, what it is that was actually happening in the active site. And I could think of two scenarios that had enabled the enzyme to become faster at this reaction. The first is that the enzyme is binding the ketone and then facilitating tautomerization to speed it up and make the product. And the other is just that the enzyme had been evolved to bind the enol when it forms and then reacts with it. This is kind of a more traditional way that enzymes work, but I didn't know if I was able to install any way to facilitate the tautomerization, so it's possible that the enzyme was just getting better at binding that enol. Now, how do we test this? Well, let's step through a thought experiment about what might happen in either case if we were to add different quantities of enzyme to the reaction. So in this first case, we could consider we have some amount of enzyme and it's making some amount of product. And if we add more enzyme in, the initial velocity of the reaction should be doubled when we add twice the amount of enzyme, and tripled when we add 3 times the amount of enzyme. So more enzymes should equal a faster initial velocity of the reaction. What about the enol binding pathway? Well, in this case, I've drawn this a little bit differently, but the enzyme is going to sit around and wait for a tautomerization event to occur, and then it's going to react with the enol when it forms, and then it's going to sit around and wait again until that next tautomerization event occurs. So if we add another enzyme in, it's just going to sit around and not be able to do anything because there's no more enol in solution. So there's a point where you can saturate it and have more enzyme than is really necessary in the reaction. And I actually did this experiment. This is a reaction progress curve where there's the amount of product that's formed over time by different concentrations of enzyme. And what I can do is I was able to extract the initial velocity of all of these curves and plot that as a function of enzyme concentration. Now, in the first case, where the velocity should linearly increase the ketone binding pathway, we would expect the behavior to look something like this slope here. But as we see, it's pretty obvious that's not what is happening. We see that the enzyme rate seems to asymptotically approach this cap of the velocity of exchange of this ketone to enol of the 2-fluoroacetophenone. So the next thing that I did is I thought, well, does the faster rate of exchange mean faster product formation? And the way that we could probe this is we could measure all of the different rate constants for a bunch of different substitutions that we know should change how fast the exchange happens, and see if there's any correlation with the total turnovers that the enzyme could do. What we find is that there's not really a correlation. For example, here, acetophenone actually exchanges an order of magnitude faster than propiophenone, but the enzyme is actually much more active with propiophenone. So there's, what I think is happening is that there's something in the active site that is recognizing this alpha position, and it's obviously not there on acetophenone. Some of these I couldn't measure because they were not soluble or hydrolyzed, but I also found that some of the products, so this really cool cyanoacetophenone, actually didn't like to sit as the amine, they sat as the enamine, and this made a really cool conjugated system, which made for a beautiful green molecule. I just thought that was interesting. And finally, and I think most importantly, I realized these products look so similar to proline that if we were to reduce them, we could reach a proline derivative. So as a proof of concept, I used sodium cyanoborohydride, and I did reduce it, and I proved that you could make substituted prolines with this. So in summary, I have expanded the tryptase nucleophile scope to include ketones. I have demonstrated that the enzyme can be engineered to intercept enolates. There's absolutely no need for highly specific modes of activation. So what I thought at the beginning was not right. Enzymes can take enolates and they don't need cool activating mechanisms. It's the kinetics and not the thermodynamics that are important for the reaction rate. And I've hopefully have set precedents for the exploration of enolate chemistry and other enzymes. And what I really hope that we can do with this enzyme in the future is use it to synthesize proline analogs with dense chiral centers, and use either chemical means or some sort of enzymes cascade to reduce all of these amines to make these chiral prolines. So this is the original substrate scope of TRPB, and I'm very happy to say that I've put myself onto this little tiny hall of fame right here with azolene and my ketones. And with that, I would really like to say thank you to everybody who has been there through my entire thesis. My committee members, thank you guys so much for your advice. My team TRPB members, well, let me start actually. Francis, thank you for having me in your lab. It's been such an honor. And Sabine, thank you so much for always being there. I really appreciate it. My team TRPB members, Andy Buller, Tina Boville and David Romney. I'm going to actually go, like Francis mentioned, I'm going to go work with them at RLS Bio, which basically it uses TRPB to make all of the tryptophans that I just showed you. And thank you, Patrick. You were an amazing co-fellow Arnold Lab TRPB grad student. Thanks, Sabine, my master's student. For the enolates project, Noah Dunham was super, super useful for all of the mechanistic work. Nick Porter, David Miller, you guys were awesome for helping me work out all of these different mechanistic problems as well. Thank you, Nat and Anders. I always love talking to you guys about everything science related. And I didn't get to talk about this project, but I had some really awesome collaborators, Chang Liu and Gordon Ricks at UC Irvine, that I wanted to thank as well, because I really enjoyed working with them. And finally, I'd like to thank the entire Arnold group. You guys made my time at Caltech very, very special, and I will never forget it. It really was the opportunity and chance of a lifetime. So, okay, now I'm gonna cry. And last but not least, I'd like to thank all of the friends that I made. Playing D&D and hanging out and going climbing and skiing it's what made grad school so, I mean, on top of the science, but it made it the time of my life. So thank you guys.
Thank you, Ella. All right, I don't know how this works. Am I the chair?
Mikhail is technically, I think.
Yes, I'm the chair. So I think we can take a few questions from the general audience before we have the closed session with just the committee. So if you have a question, just ask. Let's see if that works. And if it becomes too rowdy, then we'll have more organized mechanisms. Just unmute yourself and ask.
And maybe you can stop sharing the slide so we can see each other for the discussion. Great.
Yeah, or you can raise your hand and I can call on you if you'd like to do that. Who's got a question?
So while we're waiting for a question, Andy, I'd like to introduce you to the committee for those who didn't get to meet you while you were at Caltech. Andy is a professor at University of Wisconsin now in biochemistry and chemistry, and he's part of the committee.
Hello, hello.
Hey.
I would have had more of a youthful glow if you saw me in the past.
You still do. Okay, so let's see. It was an extremely clear presentation, so I wouldn't be surprised if there are not a huge number of questions, but let's just give another 1 minute to see if anything pops up into people's heads.
So what are the limits of TRPB?
Well, I'm not going to be using any explosive nucleophiles in the future. No, I do think that there is going to be a limit of how I guess acidic that proton is. So there are specific, I think, molecules that are just going to be too inert and they're going to be too difficult to make any sort of nucleophile out of. So there is a limit, but I haven't found it yet.
And were you surprised at the switch in the selectivity that you saw?
I mean, 100%, right? I didn't do any selection for that. So it was just happenstance and luck that it happened that way. And it was a great day to get those results. I was like, unbelievable. It's like a perfect flip.
But would that be the first time that we saw a flip in TRPB?
I believe so. Unless David's nitroalkanes, they were all selective. And I know all of our beta branch stuff was entirely selective. So yes, I believe that is the case.
But on that flip, are you concluding that it was kind of a gradual flip that went over those generations, or it in the data it just kind of happened at with one specific mutation that accounted for most of the change?
There were 2 mutations that just flipped it. So it wasn't exactly gradual. I guess it was pretty like just 2 mutations that did it, but the mutations that I did introduce are in the active site. Actually, the last round of evolution, I had done targeted mutagenesis on an active site residue that I thought I could make more room in the active site, and I must have done something that really affected the way that ketone sat in it and affected, I think, the direction of attack of the nucleophile.
Mm-hmm.
So does it make D amino acids?
No, the center that got flipped was at the fluorine, and the center that is the actual carboxylic acid is still retained.
Do you think it's possible to make D amino acids?
That's been the dream. We've talked about it a lot, and maybe doing some sort of rational design where we mutate a lysine on the opposite side of the active site to pull off the proton on serine from one direction as opposed to the other, but we have not actually done that yet. It's been on the long list of things to try with TRPB.
So I just have an uninitiated question. So please stop me if this is ill-advised. But can you compare or understand some of the effects of the mutations that you made kind of in light of versions of TRPB that appear in other organisms? I'm assuming that it appears in other organisms, but maybe this is not true.
Do you mean can I look at the active site residues and say this mutation does or does not occur in other organisms, or?
Yeah, I was curious if like the single mutations that drastically change the active site, is that an amino acid that's actually conserved if you sort of look across all domains of life versus mutations that are
That's a really great question. Actually, I worked with my undergrad to do pretty much that, where we looked at all of the different conserved residues, and most of the residues that are in the active site are so highly conserved that you don't even need the structure. You can just look at how conserved they are and say that one's in the active site, that one's in the active site. But usually they're not mutated away to anything else because they're so important. So the mutations that I found obviously were not seen in other organisms because otherwise it would have destroyed the indole activity. But it seemed to be okay for my ketone activity, of course.
Okay, well, if there are no other questions, let's give Ella one more round of applause for her great presentation. I think that concludes the public session.
I guess
Thank you
Ella
all.
really likes this enzyme because she's going to keep working with it. She'll get rid of us, but keep the enzyme.
Nice. And for the committee, do we have a separate link for the?
Yes, we do. And
We
it
do,
just
okay.
popped up in my calendar.
Okay, great. Hopefully it'll do the same for me. See you guys there.
Thank
Should
you,
we
guys.
go there at 5 or should we go there now?
I would suggest going there now, just so we
Okay.
can.
I agree.
You guys are okay? Okay, great. Thanks.
Bye, friends.
Thanks, everyone.